For the cellular functions to take place effectively, we need enzymes. For any metabolic pathway to carry on effectively, we need enzymes. So, what are enzymes? Enzymes can be simply defined as biocatalysts. What are catalysts? Catalysts, we all know, is anything which activates or speeds up the pace with which a reaction takes place is called as a catalyst. So, the catalyst does not itself undergo any change, but it only speeds up the reaction. So, such catalysts inside the cells, biocatalysts, are your enzymes. And for every reaction to take place, we need an enzyme. So, there are some important things to learn about what these enzymes are and how they function. So, the bottom line for any enzyme function is to understand that when a substrate has to get converted into a product, this has to combine with the enzyme. So, the enzyme has an active site on it where the substrate will come and bind. The enzyme substrate complex and its kinetics are very important for the substrate to get converted into a product. In the process of this enzyme substrate reaction, we have to understand that the enzymes are very substrate specific. That means for every particular type of a reaction to take place, that enzyme is specific to that kind of a reaction, based on which we can classify enzymes into 6 important classes depending upon what is the kind of a reaction that it is catalyzing. For example, you have oxidoreductases. So, these are enzymes that are associated with oxidation reduction reactions. So, generally redox reactions are the most common kind of chemical reactions that takes place in the cells. So, oxidoreductases are an important class. Then you have hydrolases. These are enzymes which are associated with breaking down of a molecule of water. So, generally we know that in almost all the metabolic reactions there is a molecule of water that is involved. Either you get the energy by breaking it down or you use it as a medium. So, the enzymes that are associated with breaking a molecule of water are called as hydrolases. Then you have transferases. So, you need to transfer one important chemical moiety from one place to the other place or one substrate to the other substrate. You have these transferases which will work. So, oxidoreductases, transferases, hydrolases, then you have isomerases which are important to convert one isomeric form to the other isomeric form. Then you have liases which are also involved in breaking down of bonds. Then you have ligases which are there for joining up of bonds. So, you can classify these enzymes basically based upon the kind of the reaction that they are catalyzing.